Hello bachcho, let's see the question. So the question says, which of the following feature is not necessary for a cloning vector? So the four options are, first one is origin of replication, second is high copy number, third is selectable marker, and the fourth is cloning sites. So in चार options में से हमें correct option को identify करना है. So the key concept in this question is to first understand what are cloning vectors. This is an easy question. Let's begin by first understanding the key concept. So cloning vectors are used for the transmission of the gene of our interest from one organism into the host organism. So cloning vectors are basically used in recombinant DNA technology. So with the help of cloning vectors, we can produce recombinant cells. So cloning vectors are used in recombinant DNA technology for the transmission of the gene of our interest from one organism into the host organism. Now, the most common types of cloning vectors which are used are the plasmid vectors, which are the extra chromosomal DNA of the bacterial cells. Also, other cloning vectors which are used are the lambda phages. Then we have cosmids and some artificial chromosomes, that is the bacterial artificial chromosome and the yeast artificial chromosomes, are all used as cloning vectors. So these are very good examples of cloning vectors. Now, every cloning vector has certain essential features. So these features are absolutely necessary to be present in the cloning vectors. So these features include, so the first feature is the presence of the origin of replication. So every cloning vector should have its own origin of replication. So हर cloning vector का एक अपना origin of replication होना चाहिए, so that वो host organism में खुद से replicate कर सके. So it does not need to depend upon any other origin of replication, but it can replicate on its own inside the host cell. Secondly, it should have at least one restriction site. Now, this restriction site is also known as the cloning site. So हर cloning vector में at least एक restriction site या एक cloning site होना चाहिए, so that we can introduce the gene of our interest into the cloning vector. So restriction site को हम जब restriction endonuclease से digest करते हैं, तो अब उस region में हम एक gene of our interest add कर सकते हैं into the cloning vector. So that is why इस site को हम cloning site भी कहते हैं. Now, the next feature that is absolutely necessary to be present in the cloning vectors are selectable markers. So हर cloning vector में at least एक selectable marker होना चाहिए, so that we can identify whether the cloning vector has become a recombinant, that is whether it has picked up the gene of our interest. Now, other features which are not very necessary include that the vector should be small in size, so that a gene of our interest can be easily inserted into the vector, and a good vector can introduce larger genes into themselves. So now if we see the options, so the first option says origin of replication. So ये option incorrect है because origin of replication is a very important feature and it should be present in a cloning vector. So that is why this is incorrect. Second one is the high copy number. So this is the correct option because this is not a very necessary feature for a cloning vector. So a cloning vector does not have to have a very high copy number. So this is not a feature of a cloning vector. So this is the correct answer. The third one is selectable marker. So again, we saw that selectable markers are very important features which has to be present in the cloning vector. So again, ये भी गलत है. And the fourth option is cloning sites. So cloning sites are restriction sites. ये दोनों का cloning vector में होना बहुत ज़्यादा ज़रूरी है. So again, this is also incorrect. So the correct answer for the given question is option number 2. That is high copy number, because this feature is not necessary in the cloning vector. So this is our final answer. Hope you understood this well. Best of luck. Thank you.